The first video that we're going to focus on is going to be prokaryotic gene regulation. And so if you remember, prokaryotic cells, they're pretty simple and they contain very loose DNA within their cytoplasm. And it's not found in a nucleus like eukaryotic cells. And then here's a diagram of E. coli, which is what we're going to focus on when we kind of talk about gene regulation. So a little bit of background into gene regulation being studied specifically in E. coli goes back to about 1961 when Jacob and Menard, that's the last name of the two scientists, they started studying regulation patterns in E. coli. And they came up with this operon model. Now I'm going to go through a couple of terms here and then I'll show you a diagram, but it's very important that you understand these terms before continuing in the video because these terms will come up again. So we have a couple of parts of an operon. You have something that's called a regulatory gene. This is typically right before the operon. It's a part of the DNA, and it's going to code for a DNA binding protein that acts as a repressor, meaning that it's going to allow transcription to occur, producing some type of product. You have a promoter region of that DNA, and this is the area where DNA polymerase can attach. RNA polymerase can attach and start transcription. We have an operator, which is right before the structural gene. It's a small area. And if this repressor binds to the operator, then transcription can occur. So it essentially acts as a block. And then you have your structural gene. And these are all the gene, or this is the gene segment that codes for the proteins and enzymes that are being made. And so we're going to look at this more in detail. Here's a very simple diagram of an operon. We have our regulatory gene here. You have your promoter region where RNA polymerase can attach and start transcription. You have your operator, and you have that repressor, which is binding to the operator. And so if RNA polymerase attached here, it can't move forward to transcribe that gene. So the repressor will stop transcription from occurring. So there are two operon models that these guys worked with, and one was for the tryptophan operon. Tryptophan is an amino acid, and then the other is a lactose operon. So the tryptophan operon, also known as trip operon, it's going to work whether or not tryptophan is present or absent. So in the absence of tryptophan, the repressor is going to be inactive, which means that the operon is on. So if tryptophan is not available in the environment, this E. coli is going to produce tryptophan, so RNA polymerase will attach to that promoter region. There's nothing blocking it from moving across this operon because the repressor protein is inactive, and so that RNA polymerase is going to move across, and these structural genes are going to be transcribed, and then translation can occur. If tryptophan is present, meaning no tryptophan needs to be made, then the repressor is going to be active, which was created from the gene or the segment of the DNA before the operon, and then that's going to actually bind to that operator region, so the operon is going to be turned off, meaning that when RNA polymerase attaches to that promoter, it's going to be blocked, so it cannot transcribe that entire strand of DNA. The lac operon is a little bit different. And so the way that it's different is that in this situation, when the lactose is absent, the repressor is active, which means that it's going to bind to the operator region, which means that the entire operon is turned off. RNA polymerase can't attach there and transcribe the strand. Compare this to the trip operon, where if tryptophan was absent, then the repressor was inactive and the operon was on. Now, if lactose is actually present, what happens is that lactose will bind to that repressor protein, cause a conformational change, which means that it can no longer fit into that operator region. So RNA polymerase can attach to the promoter region and it can transcribe the strands, producing the different proteins and enzymes that it needs. So you should go through, look at these diagrams again, try to walk yourself through them, follow along in your textbook. And if you are struggling with those words, structural genes, promoters, operator, repressible protein, all of those things, you need to go back and look at those vocab. We're going to spend some time tomorrow in class looking at a simulation of the lac operon, and that'll give you guys some time to ask me any questions that you need.